Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Adaptive Immune Responses of Vertebrates, Part 2. When we look at the adaptive responses, we need to look at specific types of cells we refer to as lymphocytes. Lymphocytes are going to be cells, sites, that reside in the lymph nodes or the lymphatic tissues. Okay, so there's the lympho cells, hence the name. The two types of cells here that we're going to be looking at are the B cells and the T cells. We'll begin looking at the T cells. The T cells are going to come in a couple of different types. Two broad categories. First category, CD4, also known as the T helper or T subset H cells. Why do they call it CD4? Well, it turns out that this type, this class of, this group, however you want to think of it, of T cells, has a receptor and a co-receptor on its surface. The co-receptor is of such a shape, and they refer to it as a CD4. Spoiler alert, there's another type of co-receptor that they refer to as CD8s, and we'll talk about that in a few slides. The CD4s can be further differentiated into Th1, Th2, and T regulatory. Each of those, 1, 2, and the regulatory, have different jobs, different roles in the adaptive immune response. Th1, as it shows right here, activates cells, goes and activates different cell-mediated or, activates different cells and suppresses other cells. The cell-mediated, think of this as the CD8 cells, and it also can go and activate and work with some of the cells in the innate, go and hyperactivate macrophages, neutrophils, depending upon what role or what job needs to be done. Th2, the T helper type 2 cells, goes and plays with B cells. B cells, as we're going to see, are the ones that make antibodies. And finally, the T regulatory cells. At some point in time, your immune system needs to quiet down, okay? The infection's gone. Time to quiet everything down and start to roll back the inflammatory response. Because if you have a always active, always hyperactive immune response, turns out your immune system is going to start attacking you. And that's when you get the autoimmune disorders. And that is not a good thing. We'll start off here with the uncommitted T lymphocyte. The uncommitted T lymphocyte is a cell that hasn't quite learned yet what it wants to be. Okay? It's going to take an antigen-presenting cell, APC. Could be a macrophage, could be a dendritic cell, doesn't matter which. That APC is going to come and interact with that uncommitted T lymphocyte. Okay? The T lymphocyte hasn't quite learned Th1, Th2 yet. APC comes to interact, also starts produce, dumping IL-1. IL-1, type of cytokine, a specific kind known as interleukins, hence IL-1, it's the first kind. There's IL-2, 4, 6, 8, all the way up through 12, I do believe. IL-1 is going to help that uncommitted T lymphocyte choose Th1, Th2. Once it goes through and starts figuring that out, then Th1 or Th2 cell as you see, branches out and starts going and interacts with different cells, and the T cell itself, Th1 or Th2, starts producing its own interleukins, different cytokines, different chemokines, and goes and starts working with eosinophils, B cells, other macrophages, natural killer cells, the works. So the Th1, as you notice, all those innate cells are to the left there with it. Whereas with the Th2, remember that one wants to work with humoral, that's going to be more the B cells and whatnot. When it comes to the CD8, remember, the CD4 had a specific shaped thing on its surface. CD8, another one. CD8 are commonly referred to as the cytotoxic T cells. Cytotoxic, cell killer, because that's what it does. CD8 cells are killers. All they do is they go and they find infected cells, viral infected, bacterial infected, parasitic infected. They go and they kill that cell. Now I have a problem with the textbook. All the other chapters in this textbook we're using, this parasitology textbooks, are pretty well written. This one, it's got some issues. Here, it keeps stating over and over that the CD8 cytotoxic T cells are going to tell other cells to lyse. And that's the dumbest thing ever. You do not want to have a cell lyse, because think about it. Inside every one of your cells is DNA, certain proteins, certain lipids, RNA, ribosomes, tRNA, all of these things that you do not want your immune system to ever see, because you do not want to run the risk of making antibodies to your DNA, to ribosomes. Okay, that's not something that the B cells and the T cells learned, back when they were in the bone marrow. Oh, that was one of the things they were tested on. Do you recognize self? Because remember, if they recognize self, they die. They never get to leave the bone marrow. Only those B cells and T cells that don't recognize get to leave. That's not one of the things they tested against. So by having your cells lyse, pop, all that stuff gets spewed forth. Things are going to start to mount, things are going to start causing an immune reaction you don't want. What actually happens is the cytotoxic T cell goes and tells the target cell to commit suicide, also known as apoptosis. Apoptosis or apoptosis tells that cell to start chewing up its own DNA, chewing up all of its own proteins and whatnot, and then to collapse. Collapse in on itself, trapping the virus, the bacteria, the parasite within it, so that a macrophage can come in and then phagocytize the collapsed cell. Everything's cleared out. Now, when you look at this figure here, I want you to notice the T cell that's interacting with that macrophage, that antigen-presenting cell. What's interacting, if you notice right there, is the MHC, because we talked the major histocompatibility complex on the APC, is interacting with that T cell via a T cell receptor. T cell receptors, okay? They are immunoglobulin-like receptors on the surface of all T cells. Immunoglobulin-like. They kind of sort of resemble, but not quite all the way, the FAB fragment of an antibody. Remember the Y part, the arms at the ends, FAB. Turns out T cell receptors have a portion that's a constant and a portion that's variable. That variable region, just like with the antibodies, going to be variable from T cell to T cell, variable B cell to B cell. And it's variable because the amino acid sequence that makes up that variable region is going to be different. Different primary, thus different secondary, tertiary, quaternary structure. And it's that overall structure at the end of that variable region that will determine the shape of the overall end of the T cell receptor, overall end of the FAB fragment of an antibody. How that is shaped will determine what kind of antigen it can interact with. Shape dictates function. Shape dictates interactions. So, as we see here, the APC, if it can interact with the T cell receptor, MHC to T cell receptor, that T cell will get activated. That T cell will now be able to go and become Th1, Th2 completely. And then it will go through, and depending upon which it is, it will follow its role. Here in this figure, it's showing a Th2 cell. A Th2 cell that's going to go and activate B cells, because that's its job.